Hi Pradyum.
Hey.
Hey.
Yeah.
So you can take over now.
Okay. Thanks a lot.
No problem.
So I welcome all the participants from on WebEx and on YouTube to our final day of the afternoon session. I'll start by introducing myself. My name is Pradyum Devaya. I'm the current president of IVSA Bidar, and I will be representing IVSA Bidar as the moderator for today's discussion. I would firstly like to thank IVS in Nepal for putting the symposium together and for this wonderful symposium that we have been going through. I would also like to thank all the IVSA Asia MOs for being part of this great opportunity that we had. For our viewers joining for us the first time, this online symposium is organized by IVSA members that stands for International Veterinary Student Association from all over Asia, which consists of 16 lectures on various aspects of public health and zoonosis by our renowned speakers. I would like to remind the participants that there are two sessions. One was in the morning and afternoon, and today the afternoon session. This session. Before I introduce my speaker, I would like to put some instructions that if there's any doubts, please put it in the chat box and it will be told to the speaker at the end of the program. Secondly, I would like to tell you all that the lecture code for this session will be announced at the end of the lecture. Thirdly, I would also request all the participants to mute their mics and video camera, so there's no disturbance for the speaker. Now I would like to introduce our speaker. Today we have an astounding speaker with us, Dr. Arun Kharate sir. He graduated in 2006 from Veterinary College, Bidar, and has completed his master's in prevalence of feline panleukopenia in wild felines from Nanda Kanan Zoo in Orissa in 2008 at Orissa Veterinary College in Bhubaneswar, that's in India, and completed his PhD in 2019 at Veterinary College, Bidar itself. To his credit, Sir has published 21 research articles both in national and international journals and presented 6 papers both in national and international conferences and was awarded with the best presentation in the 20th International Conference on Veterinary Science, Medicine and Pathology and also the best poster award on emerging animal health for better livestock production under the WTO regime. Presently, Sir is associated with Veterinary College Bidar and is serving as the Assistant Professor in Department of Veterinary Public Health and Epidemiology. It is our pleasure to have you with us on this discussion, sir. Now I would like to request Arun Kharate, sir, to present over. Sir, please could you present your webinar? Thank you.
No, this way. Am I audible?
Yes, sir. You can proceed, sir.
Okay, then I will proceed. Thank you. Hello, everyone. These are the objectives of my presentation. That is, what is the antimicrobial resistance, why it is a matter of concern, and how exactly it works, the mechanism of resistance to the antibacterials, and what is the Indian scenario, and how to contain it, what are the strategies to follow, and treatment of some important resistant bacterial infection, how to introduce. Throughout the history, there has been a continuous battle between human beings and a multitude of the microorganisms about the infection and this uh, treatment. Okay, and if you look into the history, so if you look into the history, in 1945, Nobel Prize lecturer Fleming Alexander told that it is not difficult to make microbes resistant to penicillin by exposing them to concentrations not sufficient to kill them. The same thing has actually happened in the body. Means the dose of the microorganism which is not able to kill them will make them resistant. If you look into this, the timelines of the antimicrobial resistance of this very important organism, that is TB, causing organism Mycobacterium tuberculosis. The first drug was developed in 1850, then 1882, then in 1921. Continuously, drugs, vaccines continuously well developed, but still this bacteria, which is very hard, which is very it is it has developed antimicrobial resistance over a period of some decades. Earlier, they used to call multi-resistant bacteria. Then they called extremely drug-resistant bacteria. Then in the year somewhere in 1991, that DOTS was introduced, that is directly observed treatment system. So this is how the antimicrobial resistance is a very important, and it is a matter of concern. Why it is matter of concern is because the resistant bacteria lead to treatment failures. Treatment failure. Treatment will not be successful and the mortality will be increased, and resistant bacteria may spread in the community. And low level resistance can go undetected and unnoticed. And it is an added threatens to return to the pre-antibiotic era, where people used to go out of the city, out of the village and live there, and it also adds selection pressure. So what is drug resistance? How you should understand it? Actually, drug resistance or antimicrobial resistance or antibacterial resistance is always meant like it is directed towards bacteria only. Although there will be resistance against the drugs in case of endoparasites, viruses like resistance to antiviral drugs, in case of fungi, and in case of some cancer cells. And according to WHO, the antimicrobial resistance or antibiotic resistance is defined as microorganisms that are not inhibited by usually achievable systemic concentration of an antimicrobial agent with normal dosage schedule and or fall in minimal inhibitory concentration range. Means a dose which is not able to, and that is sufficient to make it resistant against that antimicrobial agent, that is a antimicrobial resistance or antibiotic resistance. And how it develops? There are a lot of germs are ubiquitous, bacteria are ubiquitous, and not all bacteria or germs are of same kind. Some are sensitive, some are resistant. Like you can see here, some are resistant, like one shown in purple color. Those which are sensitive, the one who are blue color, will be killed by the antibiotic. The one which are resistant will survive and will multiply and emerge as a drug resistant bacteria. And this slide example how the antibiotic resistance spreads. One side, this is left side, it is a animal husbandry side, and right side it is the human community side. Whenever a bird animal harbors antimicrobial resistant bacteria, they spread through the food chain to to soil, through water. They spread to humans, and from humans it spread to some other humans in the community, maybe in the hospitals, maybe in the markets, maybe in other community gatherings. They will spread. This is how the antimicrobial resistant bacteria will spread in the community. And these are the some studies that the review article on antimicrobial resistant conducted in 2014. They they suspect that they think that the deaths attributable to the antimicrobial resistance by 2050 will be of this size. Like in North America, 3,17,000 people will be dying per year because of antimicrobial resistance, and in Latin America, 3,92,000, and in Africa, 41,50,000 people will be dying. In Europe, 3,90,000 people will be dying, and in Asia number is very huge, that is 47,30,000. So the resistance is a matter of concern. It is very important. This is how, again, a normal bacteria is killed by applying antibiotic, and if it is a mutant, it will not be killed, it will survive and it will start multiplication. There are some myths associated with this antibiotic or antimicrobial resistance, that you don't have to take all the medicines that the physicians has prescribed. You should take always whatever the medicines, antibiotics have been prescribed by the physician, and you should complete the course. You know, antibiotics will be having always a course like 3 days, 5 days, 7 days, sometimes 15 days. If you come to tuberculosis, it will take to 8 months. Earlier they used to talk about 3 months, 6 months, 8 months. Now they are talking up to 1 year. So you should complete this antibiotic course. And antibiotic resistance means the body is no longer responds to the drugs. No, that is not the case. And the only people who use antibiotic regularly are at the risk of developing antibiotic resistance. What does it means? Like, say your brother is having this antibiotic resistance problem. It doesn't mean that you may not develop that problem. The bacteria which has attacked him, it may gift its plasmids to the one bacteria which may be attacking you. Like, if your brother has developed that antimicrobial resistance problem, it is equally possible that you may also suffer same things. And the fourth one is, antibiotics can be used to treat cold and flu. As this cold and flu are commonly caused by viruses, here antibiotics will not work. And there is nothing that you can do to lower the risk of antimicrobial resistance. It is also another myth that if you do some cooperated work, like say among the communities, among the doctors, among the veterinary, there should be shared responsibilities, so that definitely these antimicrobial resistance problems can be reduced. And there are various mechanisms of actions of antimicrobials, sometimes also called as antibiotics. And these some attack cell wall synthesis, some will attack the cell wall integrity, DNA synthesis, and some protein synthesis, and cytoplasmic membrane. This is how the antibiotics work. And this is the timeline of developing of the antibiotic resistance and the antibiotics introduced. When the tetracyclines were introduced in 1950, the resistance was seen in 1959. Introduced in 1960, and methicillin was the resistance was noticed in only 2 years. This is how from time to time the antibiotics have been used, and for every antibiotic, the microorganisms have developed their own mechanisms to come over the antibiotic actions. Antibiotic resistance, what does it mean? Some microorganisms may born resistant. Some may achieve resistant by mutation, or some have resistant thrust upon them by plasmids. Like we say, some are born great, some achieve greatness, or some have greatness thrust upon them. And if you come to the mechanism of antibiotic resistance, there are two types. Intrinsic, which is natural resistance, and second one is acquired, which is also called as genetic methods. In genetic method, there are chromosomal methods like mutations, and extra chromosomal methods that is attributed to the. In the intrinsic resistance, it occurs naturally, like there are no cell walls. Say penicillins cannot attack the mycoplasma because there is no cell wall. And second one is innate efflux pumps will be there, like drugs blocked from the entering the cells or increased export. As soon as the drug reaches inside the cell, it will be thrown out of the cell. So the drug cannot act on the bacteria. And the third one is drug inactivation, like cephalosporinase. This is an enzyme developed in Klebsiella which inactivates cephalosporins. And the acquired resistance. In acquired resistance, mutations, it refers to change in the DNA structure of the gene. It occurs at frequency of 1 per 10 millions of the cells. For example, Mycobacterium tuberculosis, Mycobacterium leprae, and methicillin Staphylococcus aureus, often mutants have reduced the susceptibility. When the mutation occurs, the antibiotics cannot act. The role of plasmids is very huge when you talk about antimicrobial resistance, because it is like if you are not having that guts, or if you are not having that courage to fight with someone, you say if your friend will come to you and talk, let us fight together, and that courage you will get. That's what the plasmid will do. Plasmid will gift the antimicrobial resistant genes to the one which are not at all resistant to the antimicrobials. Here, one cartoon you can see one bacteria is gifting the genes to the other bacteria. So what are the plasmids? Plasmids are the extra chromosomal genetic elements that can replicate independently and freely in cytoplasm. Plasmids which carry gene of the resistance, they are called as R genes and are plasmids. These R genes can be readily transferred from one R plasmid to another plasmid or to the chromosome. Much of the drug resistance encountered in the clinical practice is plasmid mediated. Mechanism of resistance to the genes transfer. How it occurs? The transfer of R genes or resistance genes from one bacteria to another bacteria occurs by three processes. One is conjugation, transduction, and transformation. And the transfer of R genes between the plasmids within the bacterium, that is between the plasmid itself and within the bacterium, it occurs by two ways, that is transposons and by integrons. I will discuss what these are. Conjugation is the main mechanism for spread of the resistance in the bacteria. The conjugative plasmid make a connection tube between two bacteria through which the plasmid itself can pass. Here in the third picture, here the bacterial conjugation. The second one is transduction. This common method, this is here the bacteriophages are involved. The plasmid DNA enclosed in a bacteriophage is transferred to another bacterium of the same species, like it is seen in Staphylococcus or in Streptococcus. This is the picture of transduction, the second one. So, transformation, it is least clinical problem. Free DNA is picked up by the environment from a competent bacteria. Mechanism of resistance genes, that is by transposons. Transposons are sequence of the DNA that can move around different positions within the genome of the single cell. You can see here in the transposomes. I am showing. I am just focus on the slide. You can see the sequence of the DNA is moving in the different positions within the genome, and the donor plasmid containing the transposons are co-integrate with the acceptor plasmid, and they can replicate during the co-integration. Both the plasmid then separate each other, and they contain the R gene carrying the transposon. This is Here is another simple picture how the transposons are transferred. See here, the resistance gene is transferred from transposon donor to the transposon acceptor like this. And mechanism of resistance genes transfer by integrons. Integron is a large mobile DNA spread multi-drug resistant. Each integron is packed with multiple gene cassette, each consisting of resistant genes attached to small recognition site. These genes encode several bacterial functions, including resistance and virulence. And there are some, apart from this genetic factors, some biochemical mechanisms of antibiotic resistance are there, which play a very major role. How they play is they prevent the drug bacterium, and they modify and protect the target site, or they use alternative pathway for metabolic growth requirement, and by producing an enzyme inactivates the antibiotic, and lastly by quorum sensing. I will discuss each. Decrease permeability. The antibiotic normally which enters the bacterial cell via porine channels. Porine channels are the small pores that are present on the cell walls of the gram-negative bacteria. These porine channels, if they are blocked, this is how it is blocked now. See, these porine channels are open and it is now new porine channels into integrons. It is the antibiotic cannot enter in the cell. And the second one is efflux pump. Efflux pump, these are the cytoplasmic membrane transport proteins. Major mechanism for the resistance in the tetracycline. Some gram-negative bacteria inhibit the plasmid-mediated synthesis of the porine which obstruct the influx of the hydrophilic penicillins. That is, for example, ampicillin. Structurally modified antibiotic target sites. When the antibiotic target site is structurally modified so that the antibiotic itself cannot bind, then again the resistance is seen. This is round and here it is square. So when there is a change in structure, the antibiotic cannot enter the cell wall. And modification or protection of the target site, like ribosomal point mutation is the target site for tetracycline, macrolides, and clindamycin. So if the target site is mutated, they cannot act. Altered DNA gyrase is there. Fluoroquinolone will act on DNA gyrase. But if the DNA gyrase is altered, cannot act. And penicillins, if the modified binding protein sites are there, that cannot bind. And in case of rifampicin, mutation in the DNA dependent RNA polymerase upon which the rifampicin acts, if there is a mutation, rifampicin cannot act and the tuberculosis continues. And antibiotic inactivation, sometimes the antibiotic itself is inactivated. Enzymes, say very simple example, penicillinase enzyme will inactivate the penicillin and the cell wall cannot be broken. And by producing enzymes that inactivate the antibiotic, the other examples are like chloramphenicol, inactivated by chloramphenicol acetyltransferase, and aminoglycosides, they are inactivated by acyl phosphoadenyl, which are gram negative and gram positive bacteria, and use of alternative pathway for metabolic or growth requirement. Resistance can also occur by alternate pathway that bypasses the reaction inhibited by antibiotic, and sulfonamide resistant can occur through over production of PABA, that is para-aminobenzoic acid. And drugs and the mechanism of action. This penicillin and cephalosporin, they act upon beta-lactamase cleavage. This is how they act on the beta-lactam ring. And aminoglycosides modification by phosphorylation and adenylation. This is how the development of resistance takes place. And if there is a modification in the acetylation, chloramphenicol cannot act. And if there is a change in receptor by methylation of the rRNA, this erythromycin cannot act. And when there is a reduced uptake of the antibiotic itself, the bacteria, like in tetracycline, the antibiotic cannot act, and active export out of the cell and reduced affinity of the enzymes, which is seen in case of sulfonamides. And quorum sensing. Quorum sensing, it is just like a social discussion. Like we say, some quorum discussion. Quorum sensing is a very important feature among the bacteria. What we can say, it is a social discussion among the bacteria. Bacteria, we think bacteria may not discuss among themselves, but it is very surprising to know that bacteria, they talk among themselves, they discuss, they will decide, they will have a meeting when to emerge, when to show that a pathogenic activity, when to express that virulence factors. This is what is called as a quorum sensing. Microbes communicate with each other and exchange the signaling chemicals called as autoinducers. These autoinducers bacteria population to coordinate gene expression, conjugation, apoptosis, mobility, and resistance. Like if number secrete some autoinducer, they will some enzyme they will secrete. It will be read by the other bacteria. Like they will think that yes, we are less in number. When they are very huge in number, they will secrete some other autoinducer, and they will read. Other bacteria will read that yes, we are good number in good number now, and let us show our virulence factors now. Let us release our virulence factors. And this, why it is named as quorum sensing? It is single autoinducer from the single microbe. It is incapable of inducing any such change. So, but when it is called which is a critical density that's called as a quorum. Threshold of the autoinducer is reached and the gene expression starts. Quorum sensing signal molecules are AHL, that is alpha hemosiderin lactose, AIP, autoinducer 2, autoinducer 3 have been identified in gram-negative bacteria, and autoinducer 2 and quorum sensing system is shared in gram-positive bacteria also. And why to inhibit this quorum sensing? It is proved to be very potent method for bacterial virulence inhibition. And several quorum sensing inhibitors molecules have been synthesized, which include AHL, AIP, and AL2 analogs. And this quorum sensing inhibitor have been synthesized and isolated from several natural extracts such as garlic extract. And quorum sensing inhibitor have been potent virulent inhibitors both in vitro and in vivo using infection in the animal models. So what is the Indian scenario? Here in India, there is a lack of community awareness and availability over the counter. You can go to the any store, medical store, and you can get the antibiotic. And there is a lack of awareness about the antibiotic, and there is an absence of central monitoring and of I should say the strict monitoring agency at a central, state, and various level, maybe at district and local levels. And pathogens, Vibrio cholerae, it is resistant to furazolidone, clotrimazole, nalidixic acid, tetracycline remains effective against the Vibrio cholerae. And coliforms have extensive resistance to beta-lactam antibiotic. Penicillin, fluoroquinolones show resistance to widespread gonorrhea bacteria. And alternate drugs like azithromycin and cephalosporin should be used for this gonorrhea. And syphilis is still susceptible to that is a surprise. And gram-positive cocci, streptococci, other than streptococcus pneumoniae, it is resistant to tetracycline and macrolides around in 40% of the cases, and still it is sensitive to penicillin. And Staphylococcus aureus, it is showing methicillin resistant from 50% to 100% of cases, and vancomycin resistance is also increasing in Staphylococcus aureus. And Mycobacterium tuberculosis. This Mycobacterium tuberculosis, it is annoying since decades. Time we will take. It is having some multi-drug to extremely drug resistance. We have reached now the course regimens for this mycobacterium earlier used to be 2 months, 3 months, 6 months. Now up to 1 year, they are taking extensively drug resistance TB. Additionally, acquisition of resistant to fluoroquinolone, and one of the 3 injectable second line drugs like caprimycin, kanamycin, and amikacin can be used. And steady rise in these patients. And one important bacteria, that is New Delhi metallo-beta-lactamase. This is the enzyme was seen in bacteria Klebsiella. It is called as, this enzyme is called as NGM1, which stands for New Delhi metallo-beta-lactamase, an enzyme produced by certain strains of the bacteria, usually Klebsiella. It was noticed in New Delhi. This infection, doctor were unable to treat. They treated, they used almost all the antibiotics. Almost all antibiotics they use. The enzyme is active against the compound that beta-lactam ring like penicillin, cephalosporins, and other carbapenems. And bacteria that produce the NDM1 are resistant to all commonly used beta-lactam antibiotic, including carbapenems. And New Delhi beta-lactamase, why it is a matter of concern? Because there are currently no new drugs in the research pipeline that are aimed to stop this NDM1. Today, some of E. coli and Klebsiella pneumonia are known to carriers of this gene, but the gene can be transmitted from one strain of the bacteria by horizontal gene transfer, that is through plasmid. That's why I said plasmid play a very, very important role in the antimicrobial resistance transfer and treatment. The many NDM1 strains were resistant to all antibiotic except for the colistin. And colistin is an older antibiotic that has not been used much in recent decades. Maybe that is the reason This is why this NDM1 strain could be treated, and few NDM1 strain has been sensitive to tigicillin, that is available as a Tigacil, but this agent should be used cautiously in serious infection because it doesn't achieve high level in the bloodstream, and a few strains have also been sensitive to Estronium. And the spread of NDM can be contained with within the healthcare facilities can be curbed through strict infection control measures, including patients isolation and hand washing. And after talking about all these resistance problems and how the mechanism exactly it occurs, and what is the strategy to contain the resistance now? It is not only the one side, it is a multifactorial. Like we have to develop new antibiotics, and also we should use judiciously the available antibiotics. Like new antibiotics development, only 15 antibiotics of 167 under development had a new mechanism of action with the potential to combat multi-drug resistance. What it meant to say is it is very difficult to develop antibiotics with a new mechanism of action, and there is a lack of incentive for a company to develop antibiotics. This is the timeline of antibiotic development, and hope is not exhausted yet. They are a long way off as the research is still. And say, what are the alternative approaches like fast therapy? Can you use fast therapy? Like American countries, they are already using this fast therapy. Fast therapy is a therapeutic use of lytic bacteriophage to treat pathogenic bacterial infections. Like we have to use bacteriophages. Bacteriophages are the viruses that are very specific at the strain level against the bacteria. So we have to isolate these phages, and we have to grow in the laboratory, and in a capsule form we have to give to the patient, that you have to eat bacteriophage so that it will kill the bacteria in your body. Bacteriophages are the therapy is a very important alternative to the antibiotic, and the success rate was 80 to 95% with a few gastrointestinal or allergic side effects. And British studies also demonstrated that significant efficacy of the phages against E. coli, Acinetobacter species, Pseudomonas, and Staphylococcus aureus. And some efflux pump inhibitors can also be used. And what is the difficulty in using fast therapy in our country? I should share you, because to maintain that cold chain, it is very, very difficult to maintain that cold chain, and to subculture those phage and to maintain those things, it is very, very difficult. Some newer antibiotics like linezolid, which targets 50S ribosome, tigecycline, which targets 30S ribosome, diptomycin, depolarization of the bacterial cell membrane it causes, and dalbavacin, it inhibits the cell wall synthesis. Telavacin, it inhibits the cell wall synthesis and disruption of the membrane barrier function, and cifteberol and ceftaroline, these are the cephalosporins, and ecloprin, this can also be used, which inhibits the dihydrofolate reductase. These are the new antibiotics. Now the question of judicious use of antibiotics: how to use, when to use, like who is the authorized to use? These are the very, very important questions we should answer when you talk about antimicrobial resistance problems. Antibiotics can only judicious use of antibiotics can only stop the antimicrobial resistance, and it cannot eliminate the possibility of antibiotic resistance is a evolutionary process. The containment of the antimicrobial resistance is a multi-pronged program, like involves all the stakeholders like veterinarians, physicians, patients, and pharmaceuticals, and the general responsibility of the community. It is there are a few factors I want to discuss in this way, like what are the drug-related factors, environment-related factors, prescriber-related factors, and patient-related factors. Environment-related factors like huge population and overcrowding. Like this is itself is a problem. Like crowding of the animals, of the human beings, will lead to antimicrobial resistance spread, and rapid spread by better transport facility. We have better transport facility which facilitates the transfer of antimicrobial resistant bacteria. And poor sanitation. If we should never practice poor sanitation, we should practice good hygiene procedures, and increase the community acquired resistance. This is another problem. And ineffective infection control programs, which we should not do it. We should effectively implement the control programs against the infections, and widespread use of antibiotic in animal husbandry, agriculture, and dedicated cleansing products. Like to produce more of poultry, to produce more of agriculture products and animal husbandry products, we are using a lot of antibiotics, which is bringing pressure of this antimicrobial resistance. And drug related factors, over the counter availability of antimicrobials, strict norms should be there, script rules and regulations should be there. Like until and unless the doctor has prescribed the drugs, antibiotics should never be sold. And counterfeit and substandard drugs, like the label will be of a very good antibiotic, but inside the content will be faulty, and it will be substandard. And irrational fixed dose combination of the antimicrobials, there should be rational fixed dose combination, and soaring use of stress. Like some poor adherence to the dosage regimens, like this happens with especially with respect to the Mycobacterium tuberculosis. When the patient start feeling that I am not having cough now, I am not having that cachexia, I am putting my weight I'm feeling good, they will stop consuming antimicrobials, they will stop consuming the antibiotics, which should never be the case. And poverty, this is another important patient related factors which adds to the antimicrobial resistance. Lack of sanitation concept, and lack of education, self medication, misconception. These are the few factors. And related factors are inappropriate use of available drugs, increased empiric poly-antimicrobial use, like overuse of antimicrobials, or inadequate dosing, and lack of current knowledge and the training. And a poor clinical practice. Clinical practices sometimes poor, they will bring antimicrobial resistance problems. They will pharmacological properties of the antimicrobials, which amplify the speed of drug resistance. Antibiotic use. Antimicrobials are overprescribed, and we should never overprescribe the drugs and available without prescription. And empirical microbial selection. Antimicrobial agent indicated on the basis of clinical findings, or is it prudent to wait until such clinical findings become apparent? Sometimes this happens with the flu. With flu also, we will get some temperature. Suddenly, we should not go and take any antimicrobials, or the doctor himself also should not prescribe any antimicrobials. You should wait until unless you will get some typical clinical findings, so that the disease can be diagnosed perfectly, and a related antimicrobial agent can be prescribed. And empirical microbial selection. Can some simple bedside test can be done to confirm your suspicion? Like, can you go for a microscopy? Can you go for simple grams, so that at least say this is a gram negative bacterial infection, gram positive bacterial infection, so that there will not be any hiccup in selecting the antimicrobial. And empirical microbial selection. Have appropriate clinical specimen been obtained to establish a microbial diagnosis? Like establishing an infection is very, very important to treat the disease. Like, what is the bacteria involved? So it will take some time to isolate, to identify bacteria. It will take some time, some 2 to 3 days. If you're going for PCR, it will take at least 24 hours. So this is also very important. And what are likely etiological agents? A doctor has to think about some environmental factors in which the patient is working. What are the likely etiological agents? Maybe with smearing, with some isolation procedures, it can help. And what measures should be taken to protect individuals exposed to the index case to prevent secondary cases? Like, we have to prevent the secondary cases. Secondary cases, we have to treat the primary cases properly. Is there clinical evidence from clinical trials or antimicrobial therapy that will confirm the clinical benefits for the patient? Like evidence-based medicine, can you use that? We have to think it. And how to go about, like a definitive treatment can be a narrow spectrum agent can be used rather than using broad spectrum agent like fluoro you are using. It is a very broad spectrum drug. Can you use only penicillin? That will be sufficient. That is a definitive treatment. And sometimes in definitive treatment, we have to club the antibiotics. Is only one agent is sufficient or combination of two agents? Like, if you want to take care of gram positive and gram negative bacteria, you can club streptomycin and penicillin antibiotics so that the both bacteria can be taken care, like beta-lactam and aminoglycosides. Extended beta-lactam penicillins and beta-lactam is inhibitors, and anti-tubercular regimens, cotrimoxazole, and other drugs. And definitive treatment, like what are the optimum dose, what is the of administration, what is the duration of therapy? These seems to be very small thing or silly thing, but these are very, very important for the infection to treat. And what is the specific test to identify the patient who will not respond to the treatment, whether you are going for the microscopy or if you are going for biochemical test or whether you are going for this antimicrobial testing. What adjunctive measures can be undertaken to eradicate the infection? Are giving vaccines, steroids you are giving, or a drainage of the pus? Sometimes what happens is people will be simply treating, but there will be pus. You need to drain that pus, and sometimes you have to carry the amputation of the foot, limb, or the tail, or removal of setter, so that the infection bacterial load can be reduced. And whose work? Microbiologist should look into the bacterial sensitivity and find out the possible cause of development, and physician and or veterinarian has to treat the infection. We should not fight, like microbiologists are at a higher position and vets or physicians are at lower position, or vice versa. We are having the equal responsibility of treatment, and treatment options for selected of highly resistant bacteria are like MRSA methicillin resistant, vancomycin resistant. This, for this bacteria you can use linezolid, daptomycin, and telavacin. For streptococcus epidermidis, you can use vancomycin plus rifampicin plus gentamicin. And for this streptomyces pneumoniae, you can use naltrexone and cefotaxime. And these are the examples of the treatment for E. coli, neutrophil, and for Klebsiella pneumoniae, imipenem, meropenem, and for Pseudomonas aeruginosa, aminoglycosides can be used. And in our hospital, antibiotics recommended, these are the first line antibiotics like penicillin, oxacillin, amoxiclav, erythromycin, and second lines are vancomycin, oflux. Here we should make what are the first line antibiotics for the gram-positive bacteria, and second antibiotics for the, it is the judgment of the veterinarian or the physician. And for gram-negative bacteria, amoxiclav will be the first line, gentamicin, cephalosporin, and amikacin. And second line drugs are imipenem, cefepime, ceftaclav, and tobramycin. And the hospital for this MRSA, vancomycin, lenazolide, sparfloxacin, etc. And these are the drugs for Pseudomonas aeruginosa, piperacin, natilimacin, etc. And what is the take-home message? Take-home message is a target definitive therapy to known pathogen. Treat infection, not the contamination. Repeat, treat infection, not the contamination. Know when to say no to the vancomycin, carbapenems, and cephalosporins. I mean to say, you should know when the higher antibiotics and the pathogen, identify it so that easily the treatment can be given, and break the chain contagion, like keep your hands clean. Doctor also should keep your hands clean. Just wipe with a spirit before touching to a new patient, and start simple bedside test like gram staining, microscopy, lot of time and labor. And microbes and vectors swim stream, and they swim faster than we do. Bacteria reproduce every 30 minutes. For them, a millennium is a compressed into a fortnight. A foot and the pace of our research must keep with them, and they will overtake us. Microbes here were on Earth 2 billion years before humans arrived, and learning trick for their survival, and it is likely that they will be here for another 2 billion years after we depart, told by Cross in 19. And this is another new strain of 2020 emerging, the strain of bacteria, like the leader bacteria is saying you are the next class of drug resistant bacteria. Human continue to abuse and overuse antibiotics, your ranks will swell. So go out there and mutate, and remember that what doesn't kill you will make you stronger. This is the secret of antibacterial resistance. And to combat the drug resistance, if you don't, there will not be any cure tomorrow. I want to thank Dean Veterinary College Bidar, Karnataka Veterinary Animal and Fishery Sciences University Bidar, ISVA Bidar, the president Pradyumna, and ISVA Association Asia. Thank you one and all.
Thank you, sir, for this wonderful presentation. It was very, very informative. We have a lot of questions coming in. I will pick some questions to ask you. Sir, first question. There are a lot of products used by humans which claim to disinfect, destroy, or kill microbes. Would such also cause AMR to develop on microbial flora? If so, should we as species stop or limit the use of such products, since we also warn against the use of too much antimicrobials in animals?
Okay, I want to clear two things. Two terminologies used. One is antimicrobials and antibiotics. Okay, antimicrobials will not be, all the antimicrobials are not antibiotics. Hello, are you getting? All the antimicrobials are not antibiotics, but all the antibiotics are antimicrobials. Okay, here chemicals usually antimicrobials, like say Dettol or phenol, what you say carbolic acid. If you use that, the antimicrobial resistance will not develop against that, but antimicrobial resistance against antibiotics. Antibiotics are usually derived from the funguses, usually from funguses, except for few antibiotics like gentamicin, it is derived from bacteria. So until and unless whether it is antibiotic, you should use in a controlled manner. If it is antimicrobial, like chemicals, like you are using sodium hypochlorite, if you are using Dettol, if you are using crisol or phenol, those are antimicrobials. those also should be used judiciously, but there is no threat of development of antimicrobial resistance against those chemicals. I hope I am clear.
Apart from the ones frequently taking antibiotics. Why is it so? If a person completes the entire course of antibiotics but frequently takes the antibiotics, will he be still susceptible to AMR?
Pradyumna, can you please repeat the question, please? I am not getting.
Yes, sir. Sir, the first part of the question is who are susceptible to AMR apart from the ones frequently taking antibiotics?
Let me clarify that. Let me clarify that. Each and every one are susceptible to antibiotics apart from whosoever is using the antimicrobial antibiotics in our community. Say in your family only your only parents are using antibiotics, means all of your family members are equally susceptible to the antimicrobial resistance.
Okay, sir. So the second part of the question is if a person completes the entire course of antibiotics but frequently takes the antibiotics, will he be still susceptible to AMR?
No, no. If you are taking complete course of this antibiotics, there will not be any chances of development of antibiotic resistance. Let me say why. Because antimicrobial resistance only when you are giving the some sub concentration. Okay, like you are very reduced dose you are giving, then only the antimicrobial resistance will develop. When you are giving a complete dose, uh, it is a lethal one, then the antimicrobial resistance will not develop. It will develop only when sublethal dose is given.
Okay, okay, sir. Sir, another question. What is the benchmark to decide whether we need to prescribe antibiotics to be applied till it's finished or only take when needed? Example, an infection has healed as a way to avoid AMR. For example, when applying antibiotic cream for cat's eye infection. How likely does topical antibiotic cause AMR?
I think like whenever there is a wound, you can prescribe antibiotics. Question should arise when there will be general symptoms will be there, having cough, or you are having fever, or you are having some other problems like appetite, loss of appetite is there, or some body pain is there. In such things it will become doubtful whether to prescribe antibiotic or not. But in case of wound, okay, in case of wound, you can prescribe antibiotic, you can apply antibiotic. Otherwise what will happen is it will lead to infection with some other bacteria and funguses if you are not applying the antimicrobial.
Okay,
Is
sir.
it clear?
Yes, thank you, sir.
uh,
Sir, can NKM1 be controlled by a combination of colistatin, azithromycin, and tigecycline to overcome the challenges faced by giving a single drug?
yes, actually, this is a very big headache for the doctors. That's what I discuss. This NDM1 can be controlled with those drugs, but still the research is under going. They are conducting research continuously to develop new drugs for this NDM1.
Uh, there are reports of the presence of carbapine and colistatin resistance bacteria in animals, even though these antibiotics are not commonly used in animals for treatment. How this happens and how can we prevent the emergence of such dangerous antibiotic resistant bacteria?
This is very good question, I should say. You know what happens? See, the resistance doesn't, yeah, as like I discussed during my the classification of antimicrobial resistance. Sometimes it will be native. Okay, you can see n number of resistant bacteria have been isolated from the virgin soils. Bacteria have never been exposed to such antimicrobials or antibiotics, but they have shown the presence of resistant genes. This is how it can be present for the resistance in animals, even though it is not at all used.
Okay, sir. Sir, question. Antibiotic residue limit is defined in export animal products. So how can we meet to improve our export?
The first one is we should reduce our antibiotic usage. This is happening for the production. In our country, it is happening poultry industry, who are using huge amount of antibiotics. When it comes to the milk, we are facing some problems with the antibiotic presence in the milk, and there is no problem with the meat. I feel meat. I can say there is no problem with the meat. In the milk, how we can reduce is we should follow a strict regimen. Like withdrawal period should be observed. Like we should not use the consume milk during the course of antibiotic. This is how we can follow and reduce the presence of antibiotic in the export product.
Okay, sir. Thank you, sir. Sir, question. For treating TB, they were using antibiotics for almost 1 year. But what about the side effects of long course use of antibiotics?
Side course is like you will be acidity. This is what I have collected. This is actually experience. Some people have shared their experience with me. They are facing acidity, and sometimes they feel like they some psychological problems. Like they don't want to take the medicine continuously. They don't feel like you cannot take some 6 or 7 tablets at one time. You don't want to just gulp it. That is another problem. Some psychological factors. Otherwise, strictly speaking, medically, there will not be any problems if you consume a TB antibiotics for 1 year or more.
Okay, sir. Sir, it is clear that bacteria are resistant to antibiotics. There are different methods like conjugation, transduction, and transformation. But in
Yes.
transformation, how they get resistant gene from the environment?
See, in what happens, uh, 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 the will be picked up. The gene will be picked up from the environment. A bacteria having resistant against some bacteria against some antibiotic will die, and that DNA will be there in the environment, and that DNA consisting of the resistant gene will be picked by some competent bacteria. This is how the transformation occurs.
Okay. Okay, sir. So what can we do about the antimicrobial resistant residue in the body secretions of the animals? If we are doing mixed farming like in India, wouldn't it cause AMR as sick and healthy animals live together?
See, problem is there everywhere. So we have to use our hands, and we have to use scientific methods properly. If you, even though you are going for integrated farming, there will not be any problem unless and until you are using proper animal husbandry practices.
Okay, sir. Thank you, sir. So antibiotics are substances secreted by some microbes, usually bacteria and fungi, to restrict or inhibit the growth of other microbes. Antimicrobials comprise a broader term which includes all substances, natural or synthetic, used for restricting or inhibiting the growth of microbes. Sir, sir, how would we like to use both together in an effective method?
See, when this I told in the beginning itself, I told all the antibiotics are antimicrobials, but not all antimicrobials are antibiotics. Okay, I will give you a thumb rule. One is you should use whenever then an inanimate object is there, like a surface is there, or say some floor, or some instruments you are cleaning, some objects you are cleaning. You should use antimicrobials. Antimicrobials like Dettol, Cresol, Savlon, or say some sodium hypochlorite solution, something like that you can use. And antibiotics are used on life. Antibiotics are used for the life to treat infections. Is it clear?
Yes, sir. So can we use herbal products instead of using antimicrobials?
Herbal products, see, they also argue that we are also having a role in defeating infections. I have a little say about it. I can't say much about it because I'm not an expert in herbal medicine.
Oh, okay, sir. Thank you, sir. That was for the Q&A session. Sir, I will thank you for taking time from your busy schedule to address this important topic. The code for the today's presentation is VAR275. V for Victor, A for Alpha, R for Romeo, 275. Thank you for all the participants for attending this webinar, and I would like to hand it over to the next host for the next meeting. Thank you.
Thank you, Pradyum. Okay, so before we proceed with our next lecture, I would like to, sorry. Okay, first of all, am I audible enough? Can you guys hear me?
Ajit.
Yeah. Okay. Okay, so hi, welcome again, everyone. My name is Sajida, and I'm the exchange officer from IVSA Selangor, Malaysia. So in collaboration with IVSA Nepal and other IVSA MOs throughout Asia, we proudly present the Online Asian Symposium on Zoonosis and Public Health. So thank you so much, IVSA Nepal, for having us here today. We're really grateful for the opportunity. And okay, so basically, before we proceed, if it's your first time here, it's basically a symposium on zoonosis and public health. All right, we have various experts from various fields lined up for you guys, and we're approaching our last day. So a friendly reminder, participants can ask their doubts and queries in the chat box, which will be answered by the speaker at the end of the presentation. Lecture code will be given at the end of the lecture. Keep calm, enjoy the presentation, and jot down the code when it's given to you. Okay, last reminder, please keep your microphones and video cameras turned off and maintain decorum throughout the session. Okay, so now I would like to introduce our speaker for today. His name is Dr. Ruben Sharma. He graduated with a Doctor of Veterinary Medicine from University Putra, Malaysia, and subsequently pursued a Master in Veterinary Science from the same university. He was later awarded a PhD from the University of Cambridge, focusing on molecular genetics and phylogenomics of pathogens. Dr. Rubin has been appointed as advisor to the World Health Organization, is the director on the Executive Committee of the World Association for the Advancement of Veterinary Parasitology, and an affiliate researcher at the University of Glasgow. He has served as president of the Malaysian Society of Parasitology and Tropical Medicine, is vice president of the Veterinary Association Malaysia, a member of the National Wildlife Genetics Advisory Board, and coordinator for the Sakura Science Program Japan Laboratory at the Faculty of Veterinary Medicine at UPM, graphical distribution, molecular epidemiology, genetics and phylogenetics of parasites and zoonotic pathogens harbored by wild animals. His special interest is in zoological medicine, the ecology and conservation of Southeast Asian wildlife. Okay, so without further ado, I hand this over to you, Dr. Rubin. Sorry, Dr. Rubin, yeah, you can take over.
Hi, everybody. A very good afternoon. Already evening. It's already evening here in Kuala Lumpur in Malaysia. And I guess you guys have already had a very long day. So I will try to make my presentation as short and sweet as possible. And yes, of course, feel free to ask any questions. And if you can't hear me or if I'm breaking up, please do let me know. Yeah. And thank you so much, Sadija, for that wonderful introduction. Was pretty much a mouthful for you, but thank you so much anyways. Right, so I without further ado, I would like to proceed and share my screen. Where does it now? Okay. Right. Can you all see my screen?
Yes, doctor.
Can you see my screen? Yeah.
Yes, doctor.
Okay, great, fantastic. So I will be talking a little bit about zoonotic vector-borne wildlife diseases in Southeast Asia, in particular, and I will emphasize on something which is very important now in Southeast Asia, which is the zoonotic primate malaria, right? And I'm from the Faculty of Veterinary Medicine in University Putra Malaysia in Kuala Lumpur. Right. Okay. So before I start to present to you about wildlife diseases, I would like to just lay some foundation on how zoonotic diseases actually come about and why are they so important nowadays. So as you know, there are many biodiversity hotspots throughout the world, mostly in the equatorial region and also the Mediterranean. And as you can see, Southeast Asia is really a big hotspot for wildlife. We've got very old rainforests which supports many species and numbers of wild animals. But the sad part is that Southeast Asia is also a hub for threatened mammals. Okay, so although we've got a wonderful diversity and a beautiful rainforest, but due to various reasons, we are seeing a lot of animals, especially mammals, nowadays being threatened. And the most important threat to our wildlife in Southeast Asia is habitat loss. Okay, and of course we've got other stuff as well: overexploitation, invasive species, human disturbance, pollution, and so on and so forth. But by far, habitat loss is the biggest issue or the biggest problem that most Asian countries actually face in terms of threatened animals, be it mammals, amphibians, birds, and even reptiles. And this habitat loss is basically due to conversion of forests, conversion of wildlife habitats for human activities, for agriculture, for housing development, and also industry. And across the board, in many countries, this is the big problem. So how does habitat loss actually influence zoonotic disease transmission? It's very interesting because when you take away the natural habitat of an animal, they become displaced. They become displaced from the natural environment, and they are forced to encroach into urban areas and suburban areas, and even plantations, where they will start to colonize, reproduce very quickly, because there's abundant food, resources, and also shelter. And as you know, many people throw their garbage outside and is not properly secured. So a lot of displaced wild animals, especially the rodents, the primates, and also some birds, will actually feed on this refuse, and they grow and they reproduce very quickly. In fact, the reproduction rate of peri-domestic animals or urban wildlife are much higher than that in the natural setting. So when these animals are dispersed from the natural habitat, they often bring with them zoonotic pathogens. And then, of course, the vectors which feed on their blood will also follow them out of the forest into the urban environment. And this effectively narrows the gap between the human wildlife interface, and that, of course, allows for very efficient transmission of zoonotic pathogens from animals to humans, right? Now, this is a very interesting diagram showing you how pathogens spill over from the salvific cycle, which is the cycle of pathogens in wild animals in the forest, how it spills over to the peri-domestic wildlife. Peri-domestic wildlife are essentially wild animals which you find around human inhabitations. Okay, they can be primates, they can be rodents, they can be birds, they can also be bats, and even some reptiles as well. And then this peri-domestic wildlife actually act as a very efficient bridge to bridge the pathogen flow from the wild animals into humans and also to domestic animals. And more often than not, the salvific cycle is in equilibrium. But every now and then, either due to habitat destruction or some natural forces, there's a spillover of pathogens from wild animals into the peridomestic and the domestic cycle. So as you know, I'm sure there are many routes of transmission for zoonotic infections. It can be by aerosols, oral, by ingestion, direct contact, fomites. But today I'll be focusing more on the pathogens which are transmitted by vectors. And in this instance, they are mostly going to be arthropod vectors. Okay, arthropod meaning ticks, fleas, flies, mites, mosquitoes, and things like that. So we'll be looking at how these arthropod vectors can effectively transmit zoonotic pathogens from wild animals to humans. When we talk about zoonotic disease transmission, it's always very important that we understand the epidemiology of infection. You all are probably familiar with the epidemiological triad, which is the agent, the host, and the environment. But I like to put in the vector as a quartet because I believe that the vector the variables are very important for effective disease transmission. These four factors actually work in orchestration. So if you want to understand the epidemiology of zoonotic infections, you must understand the agent, you must understand the host, the environment, and also the vector, right? This is very interesting because it's been reported that over 60% of a human emerging infectious diseases are of zoonotic origin. And out of these, almost 72% originate from wildlife. Then, of course, there's a whole range of pathogens that wildlife can harbor from bacteria, viruses, protozoa, fungi, and also helminths. And this is very interesting because if you would like to address emerging infectious diseases in humans, you have got to understand that they originate mostly from wild animals. And in fact, if you look at a lot of the EIDs today, many viruses and protozoa and prions and rickettsias and bacteria, they originate from wildlife, and you've really got to understand this in order to alleviate the infectious disease problem in humans. Okay, so vets play a very important role in this sense, and this is actually where the One Health concept can come into play, whereby wildlife ecologists, veterinarians, medical doctors can get together to solve a common issue, which is human EIDs. Okay. Now, as you can see in this global map, Asia is really a hotspot for emerging zoonotic infections. We've got a whole range of pathogens coming from Asia, and therefore we should be on the ball about these infections which are coming from our region. I've quickly put together a simple table to show you the various taxa of animals and the pathogens that they can potentially transmit to humans. As you can see, the rodents are very high on the list. They can transmit a whole range of pathogens. The primates are up there as well, together with the bats. Okay, and as you go down further towards the reptiles and the birds, amphibians, and fish, you generally find less pathogens which are zoonotic. Now, this is a table again of just a very simple snapshot of the zoonotic viruses that can be transmitted from animals to humans, especially wildlife. And you've got in the first column the diseases or the organisms, and then the wildlife natural host, and also the mode of transmission. So quite a number are actually arthropod-borne in terms of the viruses. And for the bacteria, a lot of bacteria are not really vectored by arthropods, apart from the ricketsias. And they are basically in the rodents and the other small mammals, and they are transmitted mainly by ticks or the larger biting arthropods. Then, of course, you've got the other bacteria, which are more fecal, oral, or waterborne transmission. If you look at the parasites, a lot of the parasites are actually not really vector-borne. A lot of them are waterborne and by fecal oral route of ingestion. But one very important disease called malaria, which is endemic to Asia and Southeast Asia, especially zoonotic malaria, caused by Plasmodium knowlesi and also Plasmodium cynomolgi. The natural reservoir hosts are the primates, and this malaria is transmitted by the Anopheles mosquitoes. And today I would like to focus more on this zoonotic malaria because it is really up and coming problem in Southeast Asia, and it's also a problem for tourism. It's an issue in travel medicine as well. So in terms of One Health, malaria, zoonotic malaria is a very good model to study where people in various fields can get together. So a little bit about malaria. As you know, I'm sure, this is just a quick recap of your parasitology. Malaria is caused by the genus Plasmodium. Now, the genus Plasmodium is very cosmopolitan, okay? It can infect a wide array of vertebrate hosts from amphibians, birds, reptiles, and of course mammals as well. So you find Plasmodium in almost every taxa of vertebrates, and they are normally transmitted by mosquitoes, and in zoonotic malaria, especially by Anopheles and Culex. Now, there are 4 species which commonly infect humans. These are the traditional Plasmodium species, Falciparum, Vivax, Malariae, and Ovale. And they have been around for, God knows, for a very long time. In fact, they evolved probably even before humans were around. And so these species have effectively switched hosts from their animal reservoir to humans, and now they are considered as human parasites already. But interestingly, Plasmodium knowlesi, which I'll be talking more about later, has recently been recognized as a very important zoonotic malaria species, and it's been coined as the 5th human malaria species now. So very quick historical timeline about zoonotic primate malaria in Southeast Asia. This issue of zoonotic malaria has been around, for a very long time. Since 1899, this German pathologist, Robert Koch, discovered for the first time a primate Plasmodia in Southeast Asia. And subsequently, in the 1930s, scientists were able to describe the zoonotic pathogen, and it was first found to infect long-tailed macaques in Malaysia, and they were able to isolate the pathogen and experimentally infect humans. And this is very interesting because you can only get away with experimental human infections in the 1930s, I guess, because you probably can't do it now. But they effectively showed that the primate parasite can infect humans. And then further down the line, in about the 1960s, the first natural case of P. knowlesi infection was discovered. And in 2004, there was a big outbreak of Plasmodium knowlesi in Sarawak. Sarawak is a Malaysian state on the island of Borneo. And the authors showed that almost 60% of the human malaria infections in Sarawak were due to zoonotic Plasmodium knowlesi. And now, in 2019, we know that Plasmodium knowlesi is very widespread in Southeast Asia. And in fact, in Malaysia, it is now the most common form of human malaria and the most fatal form of human malaria in humans. Okay. So primates in Asia can harbor many species of Plasmodium, out of which 3 have been recognized to be zoonotic or potentially zoonotic. Of course, knowlesi, as I mentioned, is already confirmed zoonosis. now there's a new Plasmodium, Plasmodium cynomolgi, which has also been confirmed to naturally infect humans in Asia. And of course, Plasmodium innui, there are no natural infections yet, but experimentally, humans can get infected with Plasmodium innui. So Knowlesi and cynomolgi are very high on the list of zoonotic malaria in Asia. So this is a map of Southeast Asia showing you that Plasmodium knowlesi has been detected in almost all of the Southeast Asian countries. These dots do not just represent foci of infection, but just representation to show that the disease has been reported in animals and also in humans. Now, very interestingly, this new parasite, Plasmodium cynomolgi, has the first infection, natural infection, was detected in northern Malaysia. Subsequently, there are infections in Cambodia, in Sabah on the island of Borneo, and also some other infections in Malaysia as well. So cynomolgi is now also very widespread in Southeast Asia. Now, it is not only a localized problem in Southeast Asia, but due to a lot of travelers visiting Southeast Asia for recreation, we have actually effectively exported the disease to many parts of Europe. We've also exported it to America, and we've recently opened a branch in Australia as well. So it's not only a localized problem, but it's becoming a global problem now. And to top that up, we've also exported recently, 2 years ago, the first case of imported cynomolgi from Malaysia to Europe as well. So this is a serious emerging global infection with the foci or with the hub being in Southeast Asia. So Plasmodium knowlesi, as I mentioned, is the most common primate malaria zoonosis in Southeast Asia. It's got a very wide distribution in many Southeast Asian countries. The main reservoirs are the monkeys, specifically the macaques and the leaf monkeys. The main vectors are the forest Anopheles mosquitoes. And I'll talk a little bit about the forest mosquitoes as well, because they play a very important role in the transmission of the infection. And also how the dynamics of infection has changed a lot from being forest-specific infection now is encroaching more into the urban environment. Recently, say about maybe 5 years ago, most of the human infections can always be traced to some activity in or near forested areas. And urban infections appear to be very low. This is because the monkeys or the macaques are mostly in the forest. The Anopheles mosquitoes are very forest-specific mosquitoes, and therefore they do not breed in the urban setting. And so most cases of human infections can always be traced to activity in the forest. It may be recreation or people who go into the forest for hunting or trapping or logging and things like that. Okay. Interestingly, in the monkeys, the infection is asymptomatic as far as we can tell. Okay, and for the many monkeys that we've examined, the parasitemia is actually very low. Okay, so these monkeys are able to create an equilibrium whereby there is no overt parasitemia, and therefore they are asymptomatic. And because they do not succumb to the infection, they actually serve as very efficient reservoirs of the infection. Okay. And yes, as I said, Plasmodium knowlesi is now considered the 5th human malaria parasite. Okay, this is a graph showing you in Malaysia, okay, malaria infections since 2001 up to 2016. So as you can see, the orange bars represent the indigenous malaria, okay, that 4 classical species which we talk about, the human malaria. Then of course we got the ones in blue, which are imported cases. The one in green, the green bars represent the zoonotic malaria. So although you can see a descending trend of the total number of malaria cases in Malaysia, what you see is the zoonotic malaria is now becoming very prominent in the country. In fact, it accounts for over 70%. Actually, the latest statistics in 2019 says it's about 80 to 85% of the human malaria infections are actually zoonotic malaria. So it is a very serious problem in Malaysia. And I've also talked to my colleagues in Thailand and in Laos and Cambodia, and also on the island of Borneo. And they all agree that this zoonotic malaria is now overcoming the indigenous malaria cases in their countries. And it's something which the health authorities are very concerned about. Is this Plasmodium knowlesi a new parasite? Why is it suddenly emerging? Well, it is not a new parasite. It's been around for a very long time. The only problem is that it has been traditionally misdiagnosed, okay, misdiagnosed for other species of human malaria. For example, when you look under the microscope, and microscopy was the gold standard in the past for the detection of malaria in blood films. The trophozoites of knowlesi look very similar to the ring-formed trophozoites of Plasmodium falciparum. So microscopically, it'll be quite difficult to differentiate the two species. The band forms of knowlesi represent the band forms of P. malariae. The schizons also look like P. malariae schizons. And even the gametocytes of Plasmodium knowlesi are sometimes quite difficult to differentiate from the gametocytes of P. malariae. So in the past, medical microscopists, because knowlesi was not an issue in the past, a lot of the knowlesi infections have been misdiagnosed as either falciparum or malariae. And so now when we are looking back at all the old archival blood films, and when we do molecular diagnostics, PCR, we find that a vast majority of the old infections which were labeled as either falciparum or malariae actually turned out to be knowlesi infections, either single knowlesi infections or mixed knowlesi with other Plasmodium species. So is it a problem in humans? Yes, it is a very big problem because there are many fatal cases reported in humans in Southeast Asia. Essentially, you get hepatic and renal involvement, metabolic acidosis as well. The unique feature about knowlesi is that the parastemia can go up very high very quickly. In fact, it is one of the fastest replicating Plasmodium species in humans. And it will elicit a very high temperature as well. And in the past, actually, Plasmodium knowlesi, ironically, was used to treat neurosyphilis. Because what happens is that when the patient is infected with the treponema, which causes neurosyphilis, they would inject Plasmodium knowlesi into the patient, and Plasmodium knowlesi will elicit a very high fever, and that high fever will actually kill off the treponema, and then they would treat the knowlesi infection with chloroquine or primaquine and things like that. So knowlesi infection is very easily treated with the common anti-malarial drugs. So far, we have not seen any cases of drug resistance in knowlesi in Southeast Asia. The only thing about knowlesi is that it becomes fatal when cases are detected late in the infection. So when parastemia is already very high and there's multiple organ failure and things like that, then treatment becomes of no use, and the human patients normally succumb to the disease. So the trick about knowlesi infection is that although it's easily treated, you must pick the infection up very early in the stage of the infection. Now, as I mentioned, there's no apparent clinical signs in wild populations of macaques. The parastemia is actually very low, and this makes them very efficient reservoirs. This is an example of the long-tailed macaque over here, which is a very efficient reservoir of the infection. And then, of course, we've got the dusky leaf monkeys, which are also reservoirs of the infection as well. Sorry, excuse me, that was my phone ringing. but I decided not to answer it. Okay, so these are the lists of macaques in Asia which are known to be able to harbor Plasmodium knowlesi. Quite a number of them, mostly from the macaque group or the leaf monkey groups. Some of them are naturally infected, but in the laboratory, many other species can be infected in the laboratory, and therefore they can potentially be natural reservoir hosts as well. I will be focusing more on the long-tail macaque in today's talk. Macaca fascicularis, or the long-tail macaque, is the most abundant and the most widespread non-human primate in Southeast Asia. You can find it all over the place. It's very common. For most of you, I'm sure you've seen it it's coming to many cities and in urban settings as well. They've got a very broad ecological plasticity. They can inhabit various types of habitats. You find them from the hill forest right up to the coastal areas and the mangroves. And they are the species of a non-human primate, which is in most contact with humans. And in many places, in fact, in many forest reserves or parks, people actually feed these long-tailed macaques, and it's been introduced to many countries out of its natural range. And in some areas, it is considered as an invasive wildlife species, and sometimes even a pest. And I know the wildlife department in many Asian countries are taking steps to do population control of this macaque species. So this is the natural range of the long-tailed macaque in Southeast Asia. Of course, now it's expanded its range further up to the Asian mainland as well. But this is the natural range of the macaque. And as you can see, it overlaps with the range of the zoonotic malaria, except for northern, for example, Burma or Laos, where it is not in its natural range. But we can see we're starting to detect zoonotic malaria there already, which means that either the long-tailed macaques have expanded their range, or there may be other important primate reservoirs in these countries as well, which needs further investigation. This is a snapshot of the infection in Southeast Asia at the moment. We know that the prevalence in Sarawak, in Borneo, is very high among the monkeys. In Selangor, where I come from, where our university is at, we've got about a 30% prevalence rate, which means that almost 1 in 10 macaques would have the knowlesi infection. So it's a very serious problem. And as you can see, it's been detected in so many other countries as well. In fact, there are other countries and other prevalence rates which have been recently published this year, which have not included. But as we investigate further, we find that the infection rates and the prevalence is certainly increasing in many Southeast Asian countries. So we wanted to actually know, to study more about the epidemiology of the infection in Malaysia. So what we did was we sampled 1,500 over macaques throughout the country. We sampled macaques from various habitats, from the urban environment, the suburban environment, in plantations and orchards and agricultural lands, and also in the forests. And then we sample macaques from various age groups. We use molecular techniques to detect for the parasite, and we also did some genotyping of the pathogen as well. So interestingly, what we found in Malaysia is that the overall prevalence of Plasmodium knowlesi is slightly over 13%, which is relatively high, which means that 1 or 2 macaques out of 10 will have the infection. But more alarmingly, we found that Plasmodium cynomolgi, which is also an emerging parasite, a malaria parasite, is ranging at about 30%. This is very interesting because we know that the macaques harbor cynomolgi at a high infection rate, and we also know that natural human infections have been reported in many Southeast Asian countries. So apart from Plasmodium knowlesi, we also need to focus on cynomolgi as well. which can also be fatal. Now, this is a very interesting graph to show you that when we compare the prevalence of the parasite among monkeys in the different habitats, we found that the highest prevalence was among the monkeys in the urban and agricultural settings. This is very interesting, and as you can see, the monkeys in the forest actually had a lower infection rate. The reason I say it's interesting is because, as I mentioned to you earlier, most of the infections traditionally were always traced back to the forest. But now we have shown that the monkeys in the urban settings are even more highly infected than the monkeys in the forest. So this is something which is quite interesting, because now the infection is actually coming out of the forest and coming into the urban environment. And it follows the classical zoonotic spillover of pathogens, which I showed you earlier. And what we did was we examined the age classes, and of course, we found that the adults had higher prevalence, and also the males had a slightly higher prevalence compared to the females. Now, this data on the prevalence in the monkeys actually ties in very well with the infection in humans. Now, this is a comparative graph for 2 years only. I've got data for subsequent years, but I've not put it in. But it's showing a similar trend. So, for human infections, we can see that infections related to human activity in the forest is on a decline, whereas human infections related to activity in plantations and also in the urban environment is on the increasing. This ties in very well with our primate data to show that the monkeys are now being displaced from the forest, and they are bringing the infection out from the forest into the urban environment, and it correlates very well with the infection in humans, the trend of infection that we are seeing in humans. The only thing that is stopping the infection, the zoonotic malaria, from being a full-blown outbreak is the fact that the Anopheles mosquitoes are still very much confined to the forest. But our recent surveys, we have shown that these Anopheles are already breeding in the plantations and in the suburban environments, in the villages, in the oil palm plantations, in the rubber plantations. We're already seeing this Anopheles group of mosquitoes breeding there. And I suspect it'll be a matter of time. They will be able to adapt to breed in the urban environment. And since the infected monkeys are already present here, we will see a definite increase in the human cases once the mosquitoes are present. So in conclusion, there's a very high prevalence of P. knowlesi and Cynomolga infection among the macaques in Peninsular Malaysia, and certainly in many countries in Southeast Asia. The infection is no longer restricted to the forest, but we are seeing monkeys in the urban areas and also humans in the urban setting being infected as well at a increasing rate. We really need to focus on the epidemiology of the infection to further understand the nature of the infection and its threats in Southeast Asia. We also need to look at other primate species to see whether there's a crossover or a spillover or a switch in hosts from the macaques to the leaf monkeys, and also the gibbons and the other apes and the prosimians as well. It is a serious threat not only to the Southeast Asians or the Asians, but it's also a threat to tourists, because as I've shown you, we have imported the infections to many parts of the world already. We are definitely a hotspot now for zoonotic malaria, and the salvetic reservoir or the macaques are very abundant, and it's very difficult to control the population. And these macaques will always be a constant source for human infections as long as the mosquito vector is present. And because of this, zoonotic malaria is going to be very difficult to control, and it's going to be even harder to eliminate in this region. And this poses a very serious threat to the WHO's initiative for malaria elimination in Asia. Now, this is the map of Asia showing you areas which are at high risk of malaria. Many parts of Asia, especially Southeast Asia, either in high or moderate risk of infection. But this is only for the 4 classical human malaria parasites. This does not show you the risk for zoonotic malaria. So even just the human malarias, Asia is already at a high risk of infection. Now, and this is a very interesting map showing you that countries which are going to be malaria-free or embarking on malaria elimination by 2025. We are now in 2020 already. We've got another 5 years more. According to WHO, Malaysia should be free of malaria in 3 years. Most parts of Indochina should be free as well. And then the other parts of Asia on the progress of eliminating malaria. But because of this zoonotic malaria now, this is not going to be realized, because zoonotic malaria is a new thing and is very difficult to control as long as the reservoir monkeys are around. So the WHO initiative for eliminating malaria in Asia is going to be a very uphill task. And interestingly, there was a report in the Malaria Journal advising WHO to actually acknowledge Plasmodium knowlesi malaria as a very serious zoonosis. And it's time that they look into zoonotic malaria, because it is now the most common form of malaria in humans in Southeast Asia, especially Malaysia, and is also the most fatal form of malaria. We should embark on a region-wide surveillance program for zoonotic malaria amongst range countries to get more data on the distribution, the epidemiology, the vectors, and also the reservoir hosts. We need to understand the salviatic cycle further, and this would be a very good model for a regional approach or a regional initiative for One Health, looking into the control of zoonotic malaria in Southeast Asia. And of course, when we're talking about control, we need to always remember that we need to look into the agent factors, the host factors, the environmental factors, as well as the vector, because all these factors, they work in orchestration, and you need to be able to control every part of this orchestra in order to be able to either eliminate or to the least minimize this zoonotic infection in humans. So with that, I thank you very much. As I promised, it was a very quick presentation. I'd like to thank all my collaborators, the various universities and the government agencies. I would like to thank my students who did the sampling and most of the work in the laboratory. Most of what I presented is their work, their master's and PhD work. And with that, I thank you very much, and I would be happy to answer any questions if you have any.
Okay, Dr.
Oh yeah, sorry, just excuse me. Before I end, I would like to introduce you to a seminar or a conference which is coming up next year. It's the Federation of Asian Veterinary Associations Congress, which will be held in Kuching, Sarawak. Remember Sarawak, hotspot for zoonotic malaria? So we are going to be having the conference there, so more people can get infected. Well, the conference was supposed to be held at the end of this year. That's why our website is FAVA 2020. But as you know, because of the COVID-19 outbreak, we have postponed the conference to October 2021. But the website link is still valid. You can go to FAVA 2020 to get all the information. I hope to see you all there, because it is all the vet associations from Asia are going to be involved. Not only the vets, but also the students. There are a lot of student bodies in Asia are going to be involved as well. And I hope IVSA can also play a very important role in this conference. Maybe even taking a session to give talks. I'm organizing the conference, so do contact me if you're interested to join the conference. We've got very interesting discounted rates for students as well. So thank you very much. Hand it over to you, Sajida.
Thank you, Doctor, for the very comprehensive presentation. Okay, so for the first question, since wild animals are reservoir for several viruses and bacteria, when do they shed them? Do they shed in their usual secretion and excretion, or when they suffer from a disease?
Okay, both. Okay, when they succumb to the infection, the titers of the pathogens will normally be very high. So it can be in the body fluids, or it can transmit through aerosols or in their secretions. But of course, there are many pathogens which are in equilibrium with their wildlife hosts, which means they do not cause overt clinical signs. They do not cause a full-blown disease. But these reservoir hosts are constantly shedding the pathogens as well in the environment. So they can shed either if they're coming down with the clinical infection or not. So just like for zoonotic malaria, we know that the monkeys do not come down with the infection, but yet they can still, the mosquitoes can still pick up the pathogen and transmit to humans.
Okay, thank you, doctor. As for the next question, what is the difference between human malaria, indigenous case, and zoonotic malaria?
Okay, so when we talk about indigenous cases, we refer mostly to the classical human malaria parasites like falciparum, vivax, malariae, and uvale. These are the 4 classical human malaria parasites which have been around for a very long time. So when we talk about indigenous infections, we normally refer to them. And when we talk about zoonotic malaria, we normally refer to Plasmodium knowlesi mainly, but now also Plasmodium cynomolgi.
Okay.
Yes.
Okay, and then the next question is, as we know, primate is the wildlife host and reservoir of Plasmodium knowlesi. So is there a possibility of zoo primates to transmit the zoonotic disease to the zoo visitors, even without direct contact?
Okay, we have screened quite a number of zoo primates, and happy to say that we have not found the knowlesi infection in any of the primates yet. But of course, in zoos, there are many feral primates, the macaques around as well in the zoo setting. And these feral macaques certainly have the infection. But as I mentioned earlier, the missing link is still the Anopheles mosquitoes. We do not see the Anopheles mosquitoes in the urban environment yet. But I suspect that it will be just a matter of time. The mosquitoes will start to adapt to the urban setting, and then they would be able to transmit the infection not only to the zoo primates, but also to the zoo visitors. And then, of course, I will get more grants to do more research.
Okay, doctor. So for the next question, are there any biological control methods to control it? Could you explain?
Okay, biological control. In terms of the vector, yes, people are looking at various types of fungi, various types of microbes as well, some symbiotic microbes which can infect the mosquitoes and can kill off the larvae or prevent the mosquito from ovipositing its eggs. So these biological controls have so far been focused mostly on the mosquito vectors. But in terms of the parasite in the reservoir macaque itself, no, we are not looking at any kind of biological control for that, apart from actually controlling the population of macaques itself.
Okay, doctor. So prophylactic treatments that humans can take, are there any prophylactic treatments for humans can take to prevent malaria?
Yes, the classical chloroquine, primaquine, and of course quinine eventually can be taken to prevent malaria. These are drugs which are really advocated for travelers coming from malaria-free areas, going into malaria endemic areas like Asia, Africa, and things like that. Yes, prophylaxis is a very good way to prevent the infection, and it works very well for knowlesi infection as well.
Okay, doctor. So is there any specific reason as to why Plasmodium knowlesi is more prevalent in male macaques compared to females?
Okay, very interesting question, which I do not have the answer for at the moment. I'm working on it. I would say the simplest answer was God made it that way, but that would be it's a bit far-fetched. We believe that because the male macaques have got a wider home range, and a lot of the time they are fighting for territory, and so you get a lot of bachelor male macaques in the periphery of the harem, and these macaques are constantly fighting to get into the group, and they often undergo a lot of immunosuppression or injury and things like that. So sometimes they can be more susceptible to the infection. That is one reason. The other reason, as I mentioned, they've got a wider home range, so they're possibly more exposed to the vectors as well. But this is at the moment, this is very speculative. We cannot say for sure why they are more infected. This is certainly something which will be very interesting to investigate.
Okay, so for the next question, global warming and subsequent climate change is changing the vector distribution pattern across the world. What can we do to prevent the emergence and re-emergence of the vector-borne zoonosis that result from this?
Okay, yes, climate change, environmental change, change of land use is certainly influencing vector distribution and also numbers. It's a topic of constant debate, to be honest with you. People are trying various techniques to control these vectors, and with some success in certain areas, total failure in other areas as well. So especially when we talk about global warming, we know that a lot of the mosquitoes thrive in humid and warm environments, and we are seeing higher reproduction rates in the mosquitoes, certainly, and higher numbers as well. And in areas which used to be more or less mosquito-free, now because of global warming, we are seeing mosquitoes climbing at high altitudes, breeding at high altitudes where it used to be cold ones. Now it's becoming warmer. So they are certainly spreading not only spatially, but also in terms of going to higher altitudes as well. And also, therefore, colonizing different forest types, different habitat types as well. But to be honest, the only sure way to eliminate any zoonotic infection is to eliminate humans.
It's already happening, doctor.
Otherwise, to be honest, zoonotic infections are going to be here to stay. And the only way to get rid of zoonosis is to eliminate the final host.
Okay, so this is the next question. Why are the people of Sri Lanka at a relatively lower risk of contracting malaria?
I don't know for a fact, actually. Are they really at a lower risk? I haven't seen the data. I've not seen the statistics, so I wouldn't be able to comment on that. But if there's anybody from Sri Lanka who has some information on that, you're most welcome to contribute.
Okay. So, Doctor, Indian Veterinary Association is interested to become a partner of FAVA 2021. Would you be willing to look into that?
Oh, yeah, certainly. The Indian Vet Association is already a member of FAVA.
Okay.
They are in our council. Yeah. So I think India has 2 or 3 main associations, if I'm not mistaken. I'm not sure which one is the member, but please, whoever asked me that question, you can email me with the relevant details, and I will find out from the council if your association is a member or not. And if you are not, then of course you are most certainly welcome to join us.
Okay. So do you still have time for more questions, Doctor?
Sorry?
Do you still have more time for questions, or do you have to go?
Yeah, sure. It's 10 to 6. I can do until 6. If there are any more questions, I'm not sure if there are.
Okay, so there's one question. Is there any specific drug that's developed for Plasmodium knowlesi? If yes, then which drug?
Sorry again. What is that?
Is there any specific drug that has been developed for Plasmodium knowlesi? If yes, then which drug?
Okay, not at the moment. Some laboratories are looking into getting new drugs for malaria in general, not only for knowlesi. But because we are not seeing any resistant knowlesi cases, so most clinicians are still using the traditional anti-malarial drugs very effectively. So the pharmaceutical companies are in no urgency to develop any new drugs for knowlesi per se. But yes, I think many companies are working on new drugs, and not only new drugs, but safer drugs for malaria, because a lot of these anti-malarial drugs are very nasty to humans. and long-term use of the drugs can have some detrimental effects. So yeah, so many companies are working towards safer drugs, but for Plasmodium in general, not specifically for phenolocide.
Okay. So, doctor, our own students are asking, is FABA 2021, can they start registering now?
Yes, of course. If you register now, you will get the super early bird discount.
Or by how many percent?
Gosh, I think it's something like a 50% or 60% discount or something like that. So it's very good. Please go to the website. I can't remember the figures offhand. Please go to the website and you can get a super early bird discount. Then of course it will progress into the early bird, and then the normal bird, and then towards the end it will be the angry bird.
Okay, doctor. I think just for one last question.
Yeah.
How can we stop the effectiveness of Plasmodium?
I'm not sure what is meant by the effectiveness of Plasmodium. Can you elaborate?
Because it's a question on VBACs, and that's just the only question. Or maybe
you mean how pathogenic the Plasmodium is?
I think so.
I don't think we can stop the pathogenicity of the parasite, because this parasite has been around for a very, very long time. It has evolved with the human host, and it is still causing fatal infections. So I'm not sure if we can stop the pathogenicity of the parasite per se, but we can certainly control the infection with drugs, and also with effective vector control.
Okay, I think that's all for this session. Thank you so much for accepting our invitation and replying to all these questions. Hopefully we'll see
you're most welcome. You're most welcome, and thank you so much for having me. Very happy to share.
Thank you, doctor.
Thank you. Take care. Bye. And hope you have a good conference over the next few days.
This is the last day.
Oh, really? Oh,
yeah,
gosh. Okay.
The second last session, doctor.
Then I hope you had a good conference over the past few days.
Okay, so for our participants, I'm sorry, I forgot to read out the code. So the code for you guys is BKT997. I hope you guys get the code. Okay, so thank you, doctor. We can proceed to
thank you. Take care. Bye.
Bye. Okay, so right now I'm going to pass over to Pratik from IVSA Nepal to moderate the next session. Thank you, everyone.
Hello, am I audible?
Yes, Pratik.
Okay. Hello everyone. Namaste and Jojolapa. A warm welcome to all the participants and our honorable speakers in the final session of the entire symposium. I am Pratik Kiju, Exchange Officer of IVSA Nepal, and currently in the final year of BBSc and AS Tribhuvan University, and moderator for this session. As this is the third lecture of the afternoon session, I hope all are now accustomed with our rules and regulations. I would like to thank the organizing committee, all the 13 MOs who have been working meticulously, and also to all the participants from day one. So as this is the concluding session, I hope all will enjoy and be with us up to the last moment. For the viewers joining us for the first time, and as for the last time, this online symposium is organized by IVSA members, that stands for International Veterinary Student Association, from all over Asia, which consists of 16 lectures on various aspects of public health and zoonosis by renowned speakers from different parts of Asia, which was held over during the duration of 4 days, and now we are in the fourth day, final session of the symposium. So without further delay, I would like to welcome our esteemed speaker for the session, Ms. Jiwan Prabha Lama. She will be speaking about food safety, hygiene, and hazard analysis, and critical control point and public health. So I would like to give a short introduction about her. Ms. Jiwan Prabha Lama is former acting secretary, Government of Nepal. She started her career in government sector at the post of technical assistance in Department of Food Technology and Quality Control, DFTQC. In her long career, She became the head of department for around 30 years. As a Director General of DFTQC, she contributed most of her tenure in food-based issues like food security, research and development, quality and safety, and extension programs. She applied most of her efforts in the establishment of effective implementation of government policies and laws on food safety and nutrition securities. Furthermore, she obtained her master's degree from University of Reading, UK in food technology. As a food technologist, she has participated in numerous trainings, workshops, and conferences organized by national and international organizations. Okay, and she has presented and published numerous papers, especially in relation with to food technology and nutrition. Ms. Lama has also been recognized by Nepal government, including different organizations, for her contribution and honored with Suprabhal Jana Seva Shri, TTA Sreni Vibhushan 2015, Food Safety Activist 2014, and many more. She is a former president of Nepal Food Scientists and Technologists Association. Currently, she has been working as a senior food technology advisor at Nepal Agribusiness Innovation Center to boost up different agribusiness activities. So, Miss Zion, ma'am, are you? Can you hear me? And can you share your screen, please? Ma'am, you aren't audible. Can you turn on your mic? Ma'am, sorry, we are not hearing you, ma'am.
Are you audible now?
Yes, ma'am, you are now audible.
So I have some technical problem in laptop, so I'm more doing with my laptop. Is it okay?
Yes, ma'am. So, can I share my screen? Because I have your presentation here in my laptop.
Okay. So before presentation, I would like to thank uh, Ashirvad Pokhrel of your president of your IVSA. And I would like to thank all of your organizers. This ecosystem.
So, ma'am, can you close your laptop? So, can you use only one device to join this WebEx meeting? So,
no,
there will not be.
okay. I will close one and.
So, ma'am, can I share my screen?
Yes. It's a pleasure to share my experience in this forum. So I'd like to thank all organizers. So I'm little bit like aliens because you all are veterinarian and I'm food technology speaking in your forum. Anyway, thank you very much. I would like to share my knowledge on food safety and hygiene and HACCP and its importance. Next slide, please. Pratik, can you change the slide?
Yes, ma'am. Yes.
Second slide.
Sure,
Food technologist. We food technologists, food professional, love this statement. We are what we eat, because nowadays there's the voice of food safety is rising all over the world, in the nation and in internationally. People are arguing about the pesticide residue, veterinary drug residues, and overuse of agrochemicals, the practices used to control the food, if the standards are compliant with the SPS and TBT. So these are the voice raising by the consumers. And in the same time, if you noticed, our food our skin become repair after 30 to 35 days, and all the protein is changing. The protein is the active ingredient to change all the cells, so we become what we eat. So I think we love this statement. So if you are eating healthy food, you will be healthy, otherwise you will have faced many diseases. Next slide, please. Next slide, please. Okay, you can push. Okay. And one more, please. So consumer are asking one more. Yeah. Consumer are asking, is our food safe? So if you no, this one. Yeah, if you see the plate, it's a Nepali staple food. So consumer of Nepali, and I think in the global, they all are asking if our plates are safe. So to discuss how it is contaminated by unwanted toxics. So there I would like to show some of the studies conducted in Nepal, just some of the studies. There are many studies to understand what is the unwanted substances coming through the food in our body. Next slide, please. Next, please, Pratik. Yeah. The antibiotic residue. I was hearing about the antibiotic residue in previous presentation. It was conducted in Nepal, and what it found, the antibiotic residues were present in the meat. So it is the threat to food safety. So that plate, the meat contain antibiotic residue. What will happen if it is consumed by consumer? Next, please. Pratik.
Yes, ma'am. Yes,
And another test was conducted in chicken meat sample. If you see the sample of different bar, it looked very nice. If you see the liver portion, 100% or around 80% of sample was contained the antibiotic residue. So people love to have liver because we thought that is very nutritious. But if you check liver content, because it is deposited in the liver, in the fat tissue, all the residue is deposited. If you Pratik, can you change another slide? Yeah, if you see this picture, all the pesticide, these are colorful pesticide, are present in the liver as compared to the muscle and as compared to the gizzard. So lean meat is a little bit safer than liver. And you know, as a food technology, we always advise lactating mother, pregnant mother, children, and old people to have the liver. It's very delicious, it's nutritious, but at the same time, we are also providing them the hidden hazards of antibiotic residue. So it's a little bit concern to food safety professional. Next, please, Pratik. And another sample, there was milk was collected and tasted. It was also found 17% of sample was contaminated with the antibiotic residue. Next, please. Sorry about my technical problem. And there was a rumor in market that fish contain formalin, and after testing that, what it found is some of the fish was also contaminated with formalin. I don't know it's where it does come from. It's from water or pond, or people preserve the fish to make the fresh for a long time. The formalin was present, and it's not edible. It's very harmful for human food. Next, please. And if you see this if you see the graph, Nepal is divided in three geographical regions. One is the Himalaya, and the second one is Tarai, and third one is a plain. And in plain area, there was evidence of arsenic contamination, and there was the sample was conducted, and arsenic was tested. And in one of the district, Nawal Parasi district, it was the found arsenic is very high as compared to WHO. 100 sample was collected in this district, and 70% sample was failed to the standard. So if people are drinking that underground water, they are preparing food. So we don't know what unwanted metals come to the food through food, water, through food, from food environment, through agriculture practices. So this is worrying food safety professionals. Next, please. This is also conducted in Nepal, Kathmandu. Some of the vegetables are pesticide residues in the vegetable, and 97 pesticide were tested. Among them, 10 pesticide were found in the vegetables, and food technologists, food professionals are worried because of the toxic cocktail in the pesticide residue. There are one or more than the pesticide were present in one sample. So even another threat, the number one in the table, indosulfan. Indosulfan was banned in Nepal, but still it is detected in the vegetable. So we can pursue the consumers were raising the issue, how safe is our food? So we can pursue. We are adding the contaminants to our food. So next, please. Next, please. Next. Pratik, next, please. Next, yeah. And this is the very interesting interesting survey and analysis. So I like that very much, because previous all the analysis was done as so sample was prepared and as sample was taken. But this was done as a Nepali diet, how we consume after cooking. It was analyzed, and there is two indicator. One, if you cook properly, pesticide residue and be very less pesticide residue could found in the food. One message, and another message, the presence of banned pesticide is still their presence in the food. So it is very much a serious concern. So we have to aware the government, we have to aware the consumer. They should raise the voice of the banned pesticides should not use in the food chain. So Pratik, next please. And this is the list. Just want to know that 14 and 15 are banned already, but it's still the sample we can detect this type of pesticide residue. Next, please. Next. So I'd like to take you the journey of momo. I think all the Nepali loves momo. Most of the youngsters love the momo. They can have in the snacks, they can have in the dinner, they can have in the breakfast, not in the breakfast, daytime meal. And it's very popular in Nepal. It's like a dumpling. You can add anything, raw material, either vegetable or meat. But in Kathmandu, in other region of the Nepal, the meat, different meat are used as a chicken or mutton or pork. But in Kathmandu, it's very buff. Momo is popular. I think it's very popular in Nepal. If you take the journey, how it started, so this is the buffalo market, and you can see the first corner, left corner, the buffalo market. No veterinarian are there. Nobody check the animal health. There is just auction the animal, and it's bring to the slaughterhouse. Second one, and we had one big slaughterhouse, but it's closed because consumer does not like the frozen meat. So we had the practice of fresh slaughtered meat. So this is unhygienically slaughtered the meat. This both the primary production, we don't know what happened. Maybe it's contained high antibiotics. We don't know what it contains. And then after the lower slides, if you see the beautiful momo, if it comes in front of you, you will love to have this. And we thought that after cooking, there will be some. There is safe food. If the hot food and freshly prepared food is safe, but still there is a question, the meat is safe or not. So anyway, we love the momo. Anyway, if it comes in front of me, I will love, but we have to assure the meat should be on safe to prepare the safe momo. Next, please. Next slide, please, Pratik. Next, again. Can you again press again? Yes. So after discussion, consumer thought that our food, our plate is safe, and it was proved that the plate contain pesticide residue, veterinary drug residue, mycotoxins, and other unwanted chemicals or unwanted. It is also unhygienically prepared. So sometimes it's worried all the professional, but we have the remedies. We all can eliminate all these unwanted chemicals. So next, please. If we implement food safety approaches, it can eliminate all the unwanted hazards. So if we implement good veterinary practices, good agriculture practices, good manufacturing practices, if government if government implemented effective regulations, if it's collaborating with all the stakeholders in the chain, if HACCP is implemented, if food safety management system is implemented, if farmers start to grow organic, or the awareness to all the stakeholders, if it is possible after implementing all these food safety approaches in the food chain. Next, please. I don't want to read what is food safety, but food safety is the food will not cause harm to the consumer. As we discussed earlier, if liver contain antibiotics, it is harmful for them. The food is not safe. So while we're discussing HACCP, I think everybody know about the HACCP. What does it means? Is hazard analysis critical control point. And food safety management now is the food safety management system is popular in all over the country. Sometimes it's called ISO 22000, and it identifies, it evaluates, it controls the hazard. I will repeat, it identifies, it evaluates, and it controls the process. So I will just introduce the principle of HACCP. I don't want to go all the system steps and all these things in trees, and I don't want to discuss in this short time. But this 7 principle is like our Gita, is like our Bible, it is like a Quran. All the profession who is involved in the food safety, involved in the food processing, involved in the farm. So HACCP will conduct the hazard analysis, all the hazard. There are 3 hazards. One is biological hazard, one is chemical hazard, and third one is physical hazard. It conducts the hazard analysis in every chain, and it determines the critical control points, establish the critical limits. For example, if milk is pasteurizing, time and temperature is critical limit. It established the monitoring system, it established the corrective action, it established the verification procedures, and established the documentation. Documentation. It says all about the documentation. If there is no documentation, then it's not done. So everything should be documented and monitored and verification and implementation. Next, please. Next, please. This is the approach. So HACCP alone come in one step. So this is the farm to fork concept of food chain. So who involved in the producing, farming, or who is involved in the retailer, or who is involved in the distribution, involved in the processing sector, the restaurant or home. Everyone in the stakeholder, everybody knows about the risk. Everybody know about the hazard of their own product. Otherwise, if one actor fails to adapt the procedure, other actor cannot replace that product. So we used to say, if garbage in, garbage will be out, and the consumer plate, we can have a garbage after that. So next, please. Pratik.
Yes.
This is thumb rule. This is the thumb rule of HACCP. So all the prerequisite programs should be documented, and it's recommended to production of the safe and wholesome food. To produce the wholesome food, it should be documented. It should be written, documented. Standard operation procedures are written. If agricultural farmers are involved, they should follow the good agricultural practices, and they should always follow the hygienic practices, personal hygiene, and good manufacturing practices. And if the product goes to the industry should follow the HACCP rules and regulations and procedures to produce safe. And then consumer will be happy to have safe food in their plate. And similarly, if the actor is using the transportation or deliberation, distribution, they should has also documented code of practice. They should follow that. Even the storage, groceries, and all the markets, they should follow all the procedures. And even the restaurant people, they should follow the good hygienic practice, good manufacturing practice, and the consumer will be happy to see the safe food in their plates. So this is the thumb rule. At least, or the chains will be maintained, like our golden chain or silver chain, what we wear. All the actors will combine with one another. If one actor fails, there will no longer change remains. So this is the thumb rule of HACCP. So every stakeholder, every actor should know what he's doing and what he's following. If there is some problem, they should correct that steps and monitor and evaluate and document it. Next, please. This is the prerequisite program. All the buildings and equipment should be designed well. It is very easy and facilitated to sanitize and clean. And there is a proper place for stores all the chemicals and all these things. And there is the facilities of cold and hot water. And the personal hygiene should be maintained. Next, please. And maintenance of the equipments and buildings layout should be done in all the sectors, and waste management is written and it should be followed. There should be pest control document and follow for the pest control procedures. And supplier, if they are supplying from the farm or this, they should be approved and they have documented. And all the employee and staff should be trained. They should all the employees should know what they are doing, and the management should be committed. Likewise, ceiling, wall, light, ventilation, doors, all should be maintained and clean. If it's broken, it should be maintained and it should be documented. Next, please. Next, Pratik.
Yes, ma'am.
And like this one is personal hygiene. Personal hygiene should be very highly standard in hygienic practice. We notice that people are digging the nose and spitting everywhere. Now, because of the COVID pandemic, people are using hand washing facilities, and you started to wash their hand. Either they are using sanitizer. It should be strictly monitored. It should be strictly applied, and it should be high standard hygiene practice should be conducted. And personal employee health check must be written in that document. Next, please. The effective plan of all the cleaning it should be monitoring, and it should be well mentioned. It should be clean before and after the processing, or before after the using the machinery, before after entering in the premises. It should be inspected monitoring. It should be written in the document. Next, please. Next. So equipment, what type of equipment should be used? It should be very easily to clean in the surface should be smooth. It should be durable. It should be non-toxic, it should be non-tannic, non-porous, and non-flaking. And if equipment are using thermometer and the balance, it should be calibrated regularly. Next, please. Next. Okay, this is also thumb rule. Just to understand what are the hazards. There are 3 hazards: chemical hazard, physical hazard, and the biological. Biological hazard are pathogens, and it cause harm and illness and foodborne illness to the human body, if they inhale the pathogens. So another one is chemical hazard. There are many chemicals using in food industry. They can be food additives, they can be preservatives, they can be plant sanitizer, cleaning agents, and agrochemical fungicide, pesticide, and other thing. They are using growth hormone and naturally occurring some mycotoxin and histamine. Some has the mushroom poisoning also. This also comes under the chemical hazard. And physical hazard, Asian people, I don't know, because all participants from in Asia, but especially in Nepal, what I observed that they are not bothered about the physical hazard. If you are eating the rice and you have some stone, you just throw the stone and no bother and you eat. But it causes some problem if you eat. If you inhale these physical metals or glass or wood or something other objectionable matter. So these 3 are hazard. It should be controlled in the HACCP. But nowadays, allergens has to come. Allergen is coming in the discussion. Who has allergy after inhaling, after consuming soybean and milk and some fish? And they are also raising that voice. Allergens are also hazard, but it was not taken as a hazard. But it should be labeled in the product. It contains such product allergens, so it should be labeled, and the people can study people can look at the label and buy while they are purchasing their products. So these 3 hazards should be maintained in every steps of the chain. Next, please. Next. And this is HACCP is not HACCP is not a zero risk system. It's not zero risk system. It should be maintained. It should be maintained the risk of food safety hazards, anticipating hazard in the key prevent to them. Hazard could be integrated in the food system, applying the quality system, applying from the farm to fork approach. Next, please. Next. The issues of I think it's all we all have this type of issues. Even the underdeveloping country, they do not harmonize the act according to the international standard Codex OIE and IPPC. And implementation of food safety system is very difficult, and people are now trying. The large industry are trying to implement this type of system, and we don't have the very good laboratories. We have some of the laboratories are very good, but all of the laboratories should be strengthened, and the capacity building of that all laboratory workers on risk analysis. What are the risks they have? They should know, and they should analyze this, and they should communicate. Next, please. Next, Pratik.
Yes, ma'am. It's next.
Next slide, please. Next. I will describe a little bit about the roles of all the agencies, roles and responsibility. Government agencies should formulate the laws and regulation, and harmonize the standard, and they should comply with all the agency stakeholder capacity of this all human resource management, and it should be strengthening their stakeholders and engage the stakeholder. Otherwise, it's very difficult. Government alone cannot achieve the food safety. Next, please. Next slide, please. Yes, the private sector is the key actor in the food safety. In the food safety, maintaining the food safety, but they can. It's very small. I could not read. They can compliance the regulations, and they can implement the ISO system. The management should be committed for the food safety, and they have the capacity building in the working personnel, because their staff should understand. They should have the collaboration with the government agency as a private-public partnership. If you see the European system, the good agriculture practices are implemented by private body, and it was endorsed by government. But in Nepal, there is no private standard. Every standard should be set by the government. So they can collaborate with the government, and they can collaborate in research also, with joint research with government or donor or international agencies as well. I think another one, and they can, and the small and medium industry can learn from the large industry. So they are the key role, the key agency, key sector to implement the food safety. Next, please. Next slide. Okay, academic institution. They should upgrade and update the curriculum and focus the practical knowledge and skill. So many students pass out from the graduate or master's or PhD, but they don't have the practical knowledge. So it should be focused on practical knowledge. The curriculum should be focused on knowledge, and the academic institute has very key role also to keep the food safety in the chain, because they can network, they can link us, they can collaborate with the government, private agency to join to project, joint research, joint training. They can have this type of program. They can solve the problem activities, working with government and private sector, and competence resources can be come through this academic institution, and academic institution can solve the problem of entrepreneurs and government. So if they collaborate, So they have some roles and responsibility. You have also, you all students are here, so you have also roles and responsibility to maintain the food standard, food safety standard in country. Next, please. And consumer in Nepal, consumer association are very active. They are working on the awareness of food safety, and they are sharing the information and communication. They are focusing on the proactive activity. They should engage by the government, by the private sector, by the academia. I think it's very good to have their role in the food safety area, because they are the watchdog. They are the whistleblower. So they know all the things, and they report it to government and other agencies. So they should be capacity building. They should be capacitated. Next, please. And this I like this slide very much. This is the way how every government, every entrepreneur, every academia or consumer should work together hand in hand, and government should comply the international standards and collaborate with the stakeholder for food safety. Then food safety can achieve. I like this slide very much. When I'm preparing, I love to have, but it's very difficult to get the collaboration between the stakeholders. Next, please. I don't want to go on about the COVID and food safety, because how we know how it is transmitted. It's transmitted directly and indirectly, and the psychological threat. What I found that all the people, all the consumers, all were ringing me and asking me, do we have to consume the food during the COVID? Is it safe to have the food? So I just want to share some of the slide. Can you change the slide? Pratik, can you? Yeah. So there is no evidence. There is no evidence virus spread through food, but we should aware if it is if it is handled by infected people, if it's taught by infected people. Otherwise, there is no evidence till now the virus is unlikely to source or route of transmission of the virus. Food is not the source of virus, so we can have food. You can enjoy your food, but take care. Where do you buy the food? Where do you buy for food from? So you have to aware of that area. If there is a contaminated area, so you just eliminate that area. Don't buy from that area. But all the packet food are safe. Next, please. Next slide. Okay. So the food business industry are using preparing the food, but they should be implement the good hygienic practice. They should not work if their employer have some symptoms, if they are sick, and they should keep social distancing and on strict food hygiene and sanitation. Next, please. The same thing I was repeating. Okay, next, please. Next slide, please. Yeah, there's a key points. There were three key points. There's no evidence of COVID-19 transmitted through food, but extra care should be given for hygiene, sanitation, and health, and need to follow social distancing requirement if you buy while you're buying, or while you're purchasing, or while you're cooking. So social distancing means not absence of effective hygienic practice. Hygienic practice should be always followed. I thought that I should share some of the role of the veterinary in food safety. If we were asked to people, who should you consider for a safe meat in your plate? Any veterinarian asked to the consumer this type of question? I think you don't have. They say a cook, either or a waiter, either a family member or a farmer who produces. But people do not associate meat on the plate with vets. So they should regards, actually. They have very key role in maintaining the food safety. So their fundamental roles are both in farm and arbitrary level, and sometimes in the market as well. Next, please. Next slide, please. So according to OIE, most reported outbreaks of foodborne diseases are due to contamination of food with zoonotic agents, and often during the primary producer. So you have very good role in primary producers to keep they are safe. They don't have carry zoonotic diseases, and play a key role in investigating these outbreaks, formulating and implementing remedies of this evidence, and outbreaks will be identified. So veterinarian can work in close collaboration with other professionals. They have very good they have very key role in maintaining food safety in farm, even if they are working in the meat industry, they have key role in the industry. So they should also aware of this food safety approaches. Next, please. In the farm, at the farm level, they could work on the animal welfare medicine used for the animal feed. They can supervise the animal fluid, inspect, check, and animal and genetic disease. And in the market level, they can inspect the meat shops, and they can aware them about the genetic diseases and contamination. And the foods in the slaughter level, they can inspect the drug chemical residue, and they can inspect the antemortem and postmortem inspections also. You can see the many key role of veterinary in maintaining food safety. Next, please. And in overall strategy, they can have advice. They can advise. And one of the major issue Nepal is also facing the health certificates of food and animal products and food containing animal origins. So in collaboration, veterinarian can also use is one of the key actor in maintaining food safety. Next, please. This slide I used to say it is a golden triangle. So by discussion, we are going to conclude, and this says that food safety is a shared responsibility. Food safety is not public sector responsibility, is not private sector responsibilities, is not only academic responsibility or not only consumers responsibility. It is shared responsibility. So if all the stakeholders work together, we can achieve food safety. So it's a win-win approaches for all. Next, please. Next slide, please, Pratik. Okay, this is my last slide. I have completed. Before completion, I would like to say that World Food Safety Day was celebrated in June, 7 June this year, and World Food Safety Day is the second World Food Safety Day world is celebrating, and world and UN has the food safety is everyone's business. They have the theme. So after discussion, all the slides after discussing, I think food safety is everyone's business, our business as well. So I'd like to thank all of you. Sorry for my technical problems, so I could not share my presentation. So I request Pratik every time. Thank you, Pratik.
Sure, ma'am.
If you have any clarification or questions, I would love to have.
Sure, ma'am. We are moving towards the question and answer session. So before moving towards the question and answer session, I would like to thank you, ma'am, for providing or enlightening the main topic of our
throwing their effluent to the river, and river, if the farm is nearby the river, they contaminate. It's a natural process actually.
So that's all for the question and answer session. So thank you very much, Miss June ma'am. So we are very lucky to have you here and to share your to hear your ideas about food nutrition and public health. So the code for today's session is T7R8D1. So I repeat, T for tiger, 7R for rabbit, 8D for dog, and 1. It's T7R8D1. So that's the code. Thank you once again, Jim ma'am. So we'll now
I'd like to
move
thank you all for giving me the platform. And thank you very much for sorry and apology for the technical problem. So it has to break every time. Thank you, Pratik, for handling me since yesterday. We are trying to manage, but thank you very much anyway. Thank you. I'll be always there if you have any questions or queries relating to food safety. If you want to learn more. Thank you very much.
Okay, thank you, ma'am. So the organizing team is very much grateful to you. So we are now at the end of the whole event, the whole symposium. So I would like to request the Asia representative of IVSA Global, Mr. Andre Famanser, to give a message or suggestion regarding this symposium. And I would like to tell that Andre has been the talisman of the whole event. Yes, he's the main man for this symposium to happen. So, and then we'll have a note from Mr. Ashivad Pokrel, the current president of IVSA Nepal, and the organizing committee coordinator for this event, for this grand event, Online Asian Symposium 2020, officially. So thank you, everyone. I'll be now signing out. Stay safe, everyone. So, Andre, are you there?
Yeah.
Okay, so the mic is yours. So yeah, bye-bye.
Okay, guys. Thank you, Pratik. Thank you all of the presentation from 17 presentation. So, guys, hello again. I'm Andre Firmansyah. I'm from IVSA Asia Regional Representative. Maybe some of you might already know me from the first session. I already gave a welcoming speech. Now, sadly, I will give a closing statement for this beautiful event. So here we are, guys, at the end of our long event. On behalf of IVSA, I would like to give our utmost appreciation for all MOs in Asia, especially IVSA Bangladesh, Bidar, China, Indonesia, Japan, Kelantan, Mongolia, Mumbai, Nepal, Pakistan, Philippines, Selangor, South Korea, Taiwan, and Thailand. Thank you, guys. I have so much gratitude from our hard work for this past month. Those late night meetings, a bit of chaos were here and there that we managed to carry on or overcome, and the ups and downs, and most importantly, like what Mahima said, to know all of you. I'm so grateful to be given a chance to know you all. Thank you so much. And I hope this event, especially for all of us, will be our stepping stone towards our future collaboration as a veterinary student or also professionals in veterinarian. This is not a goodbye, everyone. So I'm sure that we will meet again. We will collaborate again somewhere someday. Thank you all, and thank you once again. Thank you. Okay. Handing over to Ashirbad.
Thank you, Andre. My greetings to IVSA Regional Representative for Asia Region, Andre Firmansyah, speakers, organizing committee coordinators, and all the participants. This 4 days of online symposium has been a great time, a great experience. We get to know different aspects of zoonosis and public health from our veteran speaker from different country in Asia region. I hope participants get plenty of knowledge from this symposium, and they will apply for better public health in future. Not only that, but we also gathered some experiences regarding preparation of the program and also regarding how to maintain the standard of the program. So this program is the result of coordination between different member organizations of IVSA in Asia region and support from different. I must thank IVSA Global for their support throughout the program and for presenting their IVSA and IFSA agreement through our program. I would like also thank IVSA Regional Representative for Asia region, Andre Firmancia, for mentoring us, for guiding us, and for acting as link between IVSA Global and IVSA AMOs between Asia, and within the IVSA AMOs within Asia. Your mentoring, your counseling for us has been great motivation, and it has led to great success of this program. Thank you, Andre. I would also like to thank IVSA ISCA for their beautiful presentation on how veterinarians or how veterinary students can contribute to awareness of the zoonotic diseases. I would also like to express my gratitude to all Organization for Animal Health OIE for arranging a beautiful presentation and for arranging the speaker. I would also like to thank our guest speakers, and all the AMOs of IVSA in Asia region, IVSA Bangladesh, China, Bidar, Mumbai, Pakistan, Japan, Selangor, Kelantan, Indonesia, Philippines, South Korea, Thailand, for their contribution, their support, and their coordination for arranging this symposium. Without their coordination, their support, and their contribution, we couldn't have done this program. So thank you to all the member organizations. It is our collaborative effort. Also, I would like to thank the technical group who has been supporting us day and night for arranging this program, especially for the IVSA Mumbai. And I would also like to thank all the hosts and moderators who supported us through the sessions, and they supported speaker and they coordinated between speaker and the participants. I am equally thankful to the publication team who created some posters, which created so much interest in participants, and we had so much registrations, almost 1,800 registrations from almost 56 countries. Thank you, publication team. I would also like to thank participants who supported us throughout the symposium, who were in the symposium, and who made the question and answer session active. And I'm sorry for some technical errors. Maybe some of you who registered still haven't the certificate link. If you don't have the certificate link received, please mail us at Online Asian Symposium that I haven't got the certificate link. We'll send you for sure. Lastly, we had two objectives of this Online Asian Symposium. One was to disperse the knowledge on various aspects of zoonosis and public health, and the other to increase the closeness between IVSA members or IVSA member organizations of Asia. So I think we have achieved the both objective. I would like to thank again all the member organizations of IVSA Asia, and then I would hope that we will have this kind of collaboration in the future. So that's all from my side. I want to officially declare the end of this symposium. Thank you for all. Stay safe, stay happy. Thank you.